Hello, everybody. My name is Mitsunori Fukuda from Tohoku University in Japan. I'm a cell biologist and study about the molecular mechanisms of membrane traffic in various cell types, including melanocytes. First of all, I'd like to thank the selection committee to choose me a recipient of Takeuchi Medal this year. It's my great honor to receive this excellent award in commemoration of Professor Takeuchi's contribution in the pigment cell research. As you may know, late Professor Takeuchi was a researcher in Tohoku University, from which I graduated with a master's degree in 1992. At that time, he taught me the development and differentiation of mouse pigment cells. This is a memorial photo taken around 30 years ago. This gentleman with the gray hair is Professor Takeuchi, and I'm here as a graduate student. Okay, let's start my awardee lecture entitled Essential Roles of Rab small GTPases in Melanosome, Biogenesis and Transport. Today, I'd like to summarize the roles of Rab small GTPases in melanosome biogenesis and transport, which we have determined in the past 2 decades. At first, I'll briefly introduce to you what is membrane traffic and what is small GTPase Rab as an introduction. In the first part of my talk, I'll show you the role of Rab27A in actin-based melanosome transport and the role of Rab3238 in transport of melanogenic enzymes. In the second part of my talk, I'd like to talk about the role of Rab7B in protein degradation of melanosomes in keratinocytes, which we have recently reviewed. Okay, let's start with the introduction. Membrane traffic is achieved by transport of membrane-wrapped substances from a donor organelle to an acceptor organelle. Membrane traffic within the cell is a crucial process for life because defects in membrane traffic often cause human diseases. For example, defects in melanosome transport cause pigment dilution in hair and skin. To properly control membrane traffic, traffic controllers must be present within the cell, and a variety of membrane trafficking proteins have been identified so far. Today, I'd like to focus on the small GTPase Rab, one of the most important players in membrane traffic. Let me briefly explain the general function of Rab proteins in membrane traffic. Rab function as a molecular switch by cycling between 2 nucleotide bound states, a GTP-bound inactive state and a GTP-bound active state. The GTP-bound active form of Rab promotes membrane traffic through interaction with a specific bonding partner called Rab effector. In mammals, around 60 Rab isoforms are present, and each member is thought to regulate specific type or specific step of membrane traffic. However, because of the large number of Rab isoforms in mammals, the exact functions of most of the mammalian Rabs still largely remain unknown. This slide shows the phylogenetic tree of the mouse and human Rab family. Among them, only 6 Rabs, shown by red, are conserved from yeast to humans. That means the presence of 6 Rabs may be sufficient for cell survival and cell growth in unicellular organisms such as yeast. As shown by blue, half of the Rabs are conserved from C. elegans to Drosophila and to humans. Such expansion of the Rab family members in vertebrates is likely to be related to the complexity of cells and tissues in higher eukaryotes. For example, melanocytes and various immune cells are not present in Drosophila or C. elegans, in contrast to vertebrates. Because of their important roles in specific membrane traffic events, dysfunctions of 10 Rab isophones have been shown to cause human or mouse hereditary diseases. Interestingly, mutations of 2 Rab genes, Rab27A and Rab38, cause pigment dilution in hair and skin. So in the first part of my talk, I'll show you the role of Rab27A in actin-based melanosome transport and the role of Rab32-38 in transport of melanogenic enzymes to melanosomes. As all of you know, melanocytes are localized at the basal layer of epidermis. In melanocytes, melanins are synthesized and packed into specialized organelles called melanosomes. Mature melanosomes are first transported within melanocytes along the cytoskeleton by membrane traffic and finally transferred to neighboring keratinocytes through dendrites of melanocytes. Transferred melanosomes are then transported to the perinuclear region of keratinocytes to form melanin cap, which is responsible for protection of the cells from harmful ultraviolet light. Therefore, melanosome transport in melanocytes and keratinocytes is an essential event for skin and hair pigmentation. Here is a movie showing melanosome movement in mammalian skin melanocytes. As you see here, melanosomes dynamically move, and their movements are readily observed even under light microscope. You can easily track movement of individual melanosomes, these black dots, from the perinucleus to the cell periphery. Melanosome transport in melanocytes is not a simple process, and it consists of at least 6 distinct steps. In response to the sunlight, melanocytes are activated, and melanin pigments are produced and stored in melanosomes. Mature melanosomes are first transported to the cell periphery by microtubule-dependent motors. Melanosomes are then transferred to actin filaments and transported to just beneath the plasma membrane by an actin-dependent motor. Melanosomes anchored to the plasma membrane are finally transferred to the adjacent keratinocyte or hair matrix cell. Disruption of either melanosome transport step would cause pigment dilution in hair and skin. The molecular mechanism of actin-dependent melanosome transport was first revealed by the genetic analysis of human Griscelli syndrome. In melanocytes from human Griscelli syndrome patients, melanosomes themselves are normally formed and matured, but they are unable to be transferred from microtubules to actin filaments and aggregated around the nucleus. By contrast, melanosomes from chocolate mice are not fully matured because of a defect in transport of melanogenic enzymes to melanosomes. In the next few slides, I'll show you how Rab27A regulates actin-dependent melanosome transport in melanocytes. The left movie shows the melanosome transport in normal melanocytes. Fast movements around the center of the cell correspond to the microtubule-dependent melanosome transport, and slow movements at the cell periphery correspond to the actin-dependent transport. The right movie is a melanocyte showing perinuclear aggregation of melanosomes, a typical Griscelli syndrome phenotype. As you see here, melanosomes are normally transported to the cell periphery along microtubules, but they are unable to be transferred to actin filaments and move back to the center of the cell. Type 2 human Griscelli syndrome is a rare autosomal recessive disorder characterized by pigment dilution in hair and skin, as well as immunodeficiency. Due to the defect in melanosome transport, patients with this syndrome show silvery hair even in childhood. In early 20s, mutation in the Rab27A gene have been shown to cause human Griscelli syndrome and corresponding model mice ashen. At that time, however, how Rab27A regulates melanosome transport at the molecular level remained unclear. In order to determine the mechanism of Rab27A dependent melanosome transport, I think the first step is to identify specific effector molecules for Rab27A, namely the downstream signal of Rab27A. Fortunately, we have identified Rab27A effectors during the course of analysis of synaptotagmin-related molecules in neurons. Why have we focused on neurons? This slide shows the analogy between synaptic vesicle transport in neurons and melanosome transport in melanocytes. As you can see, neurotransmitters are transported from the pre to postsynaptic neurons, while melanin pigments are transported from the melanocyte to the neighboring keratinocyte. Of course, the regulatory mechanism of synaptic vesicle transport and melanosome transport is different, but we found similar proteins are utilized in both transport systems. One of such proteins is synaptotagmin, an abundant synaptic vesicle protein that regulates neurotransmitter release. It consists of a single transmembrane domain and two C2 domains that bind phospholipids. By database searching, we found novel synaptotagmin-related molecules abundantly expressed outside the brain. This small red branch. We name these proteins synaptotymine-like proteins, SLP. The most striking feature of the SLP family is that all SLP members contain the SLP homology domain, termed SHD, at the N-amino terminus instead of a transmembrane domain. At that time, we thought the SHD functioned as a novel protein motif, possibly as a protein interaction site, because SHD is also found in other proteins lacking tandem C2 domains at the C terminus. We name these proteins SLAC2, SLP homolog lacking C2 domains. Until now, we have found 3 SLAC2s. SLAC2A, also known as melanophilin, SLAC2B, and SLAC2C, also identified as myelin. We have analyzed the biochemical properties of the SHD and successfully discovered SHD function as a specific Rab27-binding domain. Binding of the SHD to Rab27 is highly specific. As you can see, only Rab27A and Rab27B, but not the other 58 Rabs, interacted with SLP2A by co-immunoprecipitation assay. Similarly, SLAC2A melanophilin specifically recognized Rab27 isoforms by East2 hybrid assays. Since both SLP and SLAC2 specifically recognize the GTP-bound active form of Rab27, they are likely to function as specific Rab27 effectors. Among the 8 Rab27A binding proteins we identified, we found SLP2A and SLAC2A melanophilin abundantly expressed in melanocytes. Both proteins are indeed associated with mature melanosomes together with Rab27A and are actually involved in the control of melanosome transport. As you can see in the left, knockdown of SLAC2A melanophilin causes perinuclear melanosome aggregation, whereas knockdown of SLP2A causes peripheral dilution, especially around the plasma membrane. This slide summarizes how Rab27A and its effectors regulate melanosome transport in melanocytes. Active Rab27A is specifically localized on mature melanosomes. SLAC2A melanophilin is first recruited to Rab27A via the SHD. An actin-based motor mouse in 5A is then recruited to Rab27A SLAC2A complex on melanosome. The resultant tripartite protein complex transfers melanosomes from microtubules to actin filaments and also regulates subsequent actin-based melanosome transport. Therefore, loss of any one of the components of this complex results in the same perinuclear aggregation phenotype. After actin-dependent melanosome transport, another Rab27A effector SLP2A anchors melanosomes to the plasma membrane through simultaneous interaction with Rab27A on the melanosome and phospholipids in the plasma membrane. Our findings indicate Rab27A regulates melanosome transport through sequential interaction with 2 Rab27A effectors, SLAC2A melanophilin and SLP2A. We usually called this melanosome transport model as a home delivery service model. In contrast to the function of Rab27A in melanosome transport, Rab38, whose dysfunction also caused hypopigmentation in chocolate mice, regulates the melanosome maturation step. Initially, melanogenetic enzymes such as tyrosinase and TYRP1 are not present in immature melanosomes. They are synthesized independently of melanosomes and transported to immature melanosomes by membrane trafficking. Rab38 and its close homologue Rab32 redundantly regulate this transport process. The transported melanogenic enzymes synthesize melanin within melanosomes, and then black mature melanosomes are formed. However, the precise molecular mechanism of melanogenic enzyme transport remained elusive because specific effector molecules for Rab3238 had not been identified at that time. In order to identify a Rab3238 specific effector, we have established Rab panels, tools for systematic and genome-wide analysis of Rab proteins. We have cloned all human and mouse Rab cDNAs and produced various expression constructs, including constitutive, active, and negative mutants with 5 different tags. We also have a collection of specific siRNAs, antibodies, gaps, GEFs, and knockout clones to analyze Rab proteins comprehensively. Nowadays, we have more than 1,000 materials, which will be useful for analyzing various cell events, such as autophagy, epithelial polarity formation, melanosome transport, cilium formation, and so on. We try to identify a Rab3238 specific effector by E2 hide screening using Rab32 as a base, and successfully identified VAP, VPS9 ankyrin repeat protein as a Rab3238 specific binding protein. Here I show the domain organization of VAP. It consists of an N-terminal VPS9 domain and 2 C-terminal ankyrin repeat domains named ANCAL1 and ANCAL2. Our biochemical analysis indicated active Rab3238 specifically interacted with the first ANCAL1 domain. When we knock down endogenous VAP, signals of TYRP1 was dramatically decreased, and the amount of melanin was concomitantly reduced, the same as in Rab3238 knockdown cells. Therefore, VAP indeed functions as a Rab3238 effector to regulate melanogenic enzyme transport to melanosomes. By using the Rab panels, we have identified several regulators of melanosome biogenesis and transport steps in melanocytes during the past 2 decades as summarized here. Finally, I'd like to focus on the unknown mechanisms of melanosome transport and degradation within keratinocytes. So in the last part, I'll show you the role of Rab7b in protein degradation of melanosomes. In contrast to the melanosome transport in melanocytes, very little is known about the mechanisms of melanosome transport and its degradation within keratinocytes. One of the major reasons is the lack of a specific marker protein that can be used to visualize melanosomes in keratinocytes. Antibodies against melanogenic enzymes, such as tyrosinase and TYRP1, cannot be used because transferred melanosomes in ex vivo keratinocytes were not at all recognized by these antibodies as shown here. Actually, melanogenic enzymes are readily degraded when transferred to keratinocytes. So a novel tool for visualizing transferred melanosomes in keratinocytes is anticipated. Recently, we have accidentally developed M-Ink as a tool for efficiently visualizing black mature melanosomes. M-Ink was originated from the tail domain of KIF1C motor. As you can see here, the purified recombinant M-Ink with T7 tag beautifully and specifically visualize melanosomes by conventional immunofluorescence analysis. Although the detailed recognition mechanism was completely unknown, it can also detect purified melanochores, suggesting it directly recognized black melanin pigments. The M-Ink technology allowed us to observe spatial distribution of melanosomes in melanocyte-keratinocyte co-culture system. In XY view, it is difficult to discriminate melanosomes in melanocytic dendrites and transferred melanosomes in keratinocytes. By contrast, in XZ view, we can easily discriminate them and identify transferred melanosomes in keratinocytes. Here is a 3D movie of the melanosome distribution in the co-culture system. By using M-Ink, we first analyzed the precise subcellular localization of incorporated melanosomes in keratinocytes. Consistent with the previous reports, the incorporated melanosomes were often surrounded by LAMP1, and such late endosomal compartments were negative for other organelle markers, including lysotracker, a highly acidic degradative lysosome marker. To identify the specific Rab isoform that regulates melanosome transporter degradation in keratinocytes, we performed a comprehensive localization screening by expressing 62 different GFP tagged Rabs in keratinocytes one by one. As a result, we have successfully identified 11 candidate Rabs as shown here. We then performed knockdown and knockout screening and identified Rab7b as a prime candidate that is involved in melanosomal protein degradation. As you can see here, in Rab7b knockout keratinocytes, degradation of endogenous tyrosinase and Pmel in mature melanosomes was significantly inhibited. Of course, re-expression of Rab7b in the knockout cells clearly rescued this phenotype. We thus concluded Rab7b promotes degradation of melanosomal proteins in keratinocytes. This is a summary slide of my talk. Rab family small GTPases are key players in orchestrating melanosome biogenesis and transport in melanocytes. Rab7b promotes degradation of melanosomal proteins in keratinocytes. We would like to review the detailed molecular mechanisms of melanosome transport and degradation within keratinocytes in our future study, and to continuously contribute to pigment cell research. Finally, I'd like to thank all of my colleagues. So thank you very much for your attention.